Good day, ladies and gentlemen. A very warm welcome to the second installment of Aquaculture Innovation Webinar organized by IPI, Aqua Innovation Centre or AIC, Trendline Agri-Food and Innovation Norway. My name is Z Tiam Lin from IPI and I'll be your moderator for this keynote session. To recap, this Aqua Innovation Webinar series aims to bring together leading industry experts and researchers from Singapore, Israel, and Norway to share latest trends in aquaculture technology, and we hope to promote an exchange of ideas, best practices, and collaboration in the spirit of open innovation through this virtual platform. In our first webinar last month, we touched on the topics of meeting rising demand through new species and intensification of seaweed farming. And I'm pleased to say that the webinar has sparked conversation between our attendees and speakers to collaborate. If you have missed the first webinar, the recording is available on IPI YouTube channel or will be provided in the link in the chat window. So in today's session, welcome back. We will discuss breaking new grounds in aquaculture. For aquaculture to be sustainable, it needs to address the challenges of fish meal to reduce costs, intensification, and disease management as they are brought together in a closed environment, so that harvest remains fruitful. So while cultivating a breeding culture that is environmental friendly. So our aquaculture industry really needs to engage innovation and leverage on science and technology and adopt engineering execution to break new grounds in its practices. I'm pleased we have put together 3 exciting sessions today in the webinar to enlighten you on this topic. So following the keynote sessions, we will have one session on feeds and nutrition, and this will be followed by health and genetics in the final section. Please stay on to learn from our leading expert. Any presentation materials or slides and recording that are distributable, I think we will make available to them after this webinar. Please look out for our email instruction. Now, without further ado, to kick off our keynote session, I would like to invite Mr. Eugene To, Director, Agri-Tech at Enterprise Singapore, or ESG, to give the opening remark. ESG is the government agency in Singapore that champions enterprise development, and Eugene and his team look after the agri-food and agriculture enterprise development. And without further ado, Eugene, please.
Thank you, Dr. Z, for the very kind introduction. Good day to everyone. Today, I'd like to talk about growing Singapore's agri-tech ecosystem, and I will divide the presentation into 3 segments. One is because of the international audience. I thought it'd be useful to let everyone have a sense of Singapore's government's direction in this area. Number 2, I want to talk about some of the challenges within the industry. And then finally, we want to see how different players in the value chain can collaborate and participate. Next. Well, yeah, we'll stick with this slide. We have decided to move into 3 specific subsectors for Singapore's agri-tech system. One is what we call modern agriculture or the horticulture. Number 2 is the modern aquaculture. And finally, we want to look at alternative proteins. Just for a fun fact, just to kick off the presentation, when I first took on this portfolio, one of the interesting facts that was presented to me was the following, that to get 1 gram of meat from traditional meat sources, you need 7 grams of feed. And then when you come down to fish or aquaculture, to get the same gram of fish meat, it's 1 gram of feed. All right. And then if you go down to alternative proteins, to get the same 1 gram of proteins, it's only a fraction. Right. So I think that's a very interesting comparison given the drive towards sustainability in recent times. And I think aquaculture will continue to play that very important role. Why agriculture in Singapore, given that we are land scarce? One is we think that food production will be the next high quality, high tech sector. And actually there are quite a lot of synergies with our existing strengths, whether it's in chemicals, biotechnology, or engineering. We really want to be an international model and enabler of self-sustainable cities. One, to make sure that our population have high quality, high sustainable food. Number 2, to be able to build that strong trust between consumers and the producers. And finally, we want our technologies to be test-bedded here and potentially be exportable to the rest of the region and the world. We have a whole of government approach when it comes to supporting agriculture and aquaculture, from physically clustering to innovation capacity through R&D, as well as developing local champions with a global footprint. Now, I think we do recognize that there are challenges within the sector here, and I want to talk about what are some of these challenges that we would have to face as an industry. One, the disease risk-causing business is an area that I think we need to pay attention to. I think a lot of research has gone into the temperate type of aquaculture, but I think a lot more work will need to go into aquaculture industry involving tropical climates or tropical fisheries. And that's where the roles of IPI as well as AIC is very important. Two, we also recognize that while we promote local production in Singapore, there is a cost disadvantage given our land scarce situation and the fact that we are competing with imported seafood from around the region. And finally, I think the challenge also will be for the industry to find new ways to package their food, because fresh seafood ultimately have short shelf life and there is a relatively small domestic market. So how can we transform this sector so that we find bigger markets? We want to welcome different parts of the value chain, whether you're an investor, whether you're a corporate, or whether you're a startup, to see how you can participate in this journey with us. I'll go into each of these briefly. The investment climate here is actually quite conducive over the last few years, right? And we see that in the agri-food space, just in 2020 alone, almost $200 million has been invested here. This is the highest in Southeast Asia, from what we understand. We have different accelerator programs that ESG support and work with, and they are here to help the tech startup to be able to work with our partners in the local industry to bring the industry to the next level. So these are some of the examples. We also want to partner with corporates, right? Whether it's working with our IPI partners or AIC partners, to be able to develop deep specialist expertise. And this is very useful because I think as over time, we want to build a pool of common shared knowledge that will be relevant to our farmers in Singapore, as well as to our technological startups, so that we can then not just make the food production in Singapore productive and safe, we can also find export opportunities. We think that there is an area of disruptive solutions coming from a very high-tech industry where Singapore is being well-resourced as a landing pad to test bait, pilot, and partner with high-potential companies to grow the various segments of the business. We want to leverage on research expertise within our ecosystem to be able to push the technology frontiers and develop cutting-edge research agenda, right? And this is where the partnerships and having such webinars is very important, so that we can have greater networks and links with the rest of the world. Finally, we want to emphasize the importance of startups and entrepreneurs in this space, because there is a room for them to drive some of the innovation. And therefore, I think ESG will continue to play a very important role in supporting this area. You can see the various parts of the value chain in this slide, and we want to give the different segments a bigger push. So this is the continuation of the previous slide, where we go from MNCs to global events and global partners. Next slide, please. My final slide. In conclusion, there is a big role for the aquaculture industry in Singapore. And I think it's important that we get the international players in to work with our local partners. And Enterprise Singapore will continue to give the industry here a very big push. We are counting on different partners in our ecosystem including the Aquaculture Innovation Centre, you'll hear from Dr. Lee later, as well as IPI, where we will continue to work with them to find and match different technology solutions. And finally, we have the Startup SG, where ESG will continue to support different parts of the value chain, especially on the startups and the entrepreneur space. With that, thank you very much for listening to my presentation, and over to you, Dr. Z.
Thank you very much, Eugene. I think that paints a very good overview of the ecosystem support, particularly ESG, pushing a lot to address our 30-30 food security and vision with SFA as well. So thank you. We will now move on. Thanks for the opening remark. Now we'll move on to the keynote session with two of our keynote speakers sharing their perspective on applied and translation research, as well as new development to support this sustainable aquaculture development. Now, before I invite them on board, if you have any question for our speaker, and that will lead into the panel discussion subsequently, please submit them through the Q&A panel at the bottom of the Zoom window. Thank you on that note. Let me just welcome Dr. Lee Chi-wee, a very good friend, Center Director of Aquaculture Innovation Centre, or AIC, here in Singapore, to share about translation research in aquaculture. And without further ado, Chi-wee, Dr. Lee, over to you.
Thank you, Dr. C. Hello, everybody. I would like to share my screen right now. Is my screen okay?
Yes.
Okay, good. Thanks, guys, for coming here to this webinar. It's my pleasure to share with you what is Aquaculture Innovation Centre is doing to support this wonderful industry in Singapore. AIC, in short form, is actually part of the ecosystem established by the Enterprise Singapore to support the aquaculture industry in Singapore, and also following our players to outside Singapore as well. Now, we all know that this industry is very diverse, is cover quite a bit of stuff, and also moving very, very rapidly. And for Singapore, we started as a non-agriculture, aquaculture country to embark on this journey. It's going to be a bit of a challenge. As a result, we find that we need to engage the talent industry deeply and bring everybody on board so that we can able to move the industry up the value chain rapidly. So with that, the AIC is actually based on the consortium models. We bring in most of the Institute of Higher Learning in Singapore together, and that will allow us to tap into the resources, primarily the talent, because it's very important that to develop this industry properly, it cannot rely on the staff coming from AIC. We need to tap into the majority of the experts and talents, doesn't matter from which disciplines, and that they come in to look into the problem statements from different angles and to support the industry to move up the value chain. Now, the AIC is actually funded and supported by Enterprise Singapore, and it's very young organizations. It's less than 2 years old, but we started our journey way back and in preparations for the Singapore Food Story and to achieve the target of 30 by 30. Now, in just one slide to cover what is the functions of the AIC, and our mission, the mission is actually to close the loop in sustainable, super intensive aquaculture. Yeah, and to enhance, of course, food security and resilience. As what Mr. Eugene To mentioned, in Singapore, we just don't have much land, and the labor cost is high, and so is the water. So as a result, everything we do, it has to be very, very super intensive. In other words, with the cost be constant, only way for us to increase our productivity is to put more things into the volume, into the space that we grow the animals. So as a result, the technologies that we need and the management system and also the operating system need to be very, very highly tuned and refined so that we can achieve that kind of productivity that we're looking for. Now, we believe to do this effectively and in the shortest possible time that we need to embark on 4-pronged approach. First, we must have a research. Research has to be. And for warm water species and the uniqueness of our culture system in Singapore, due to we have limitations in a lot of resources, and then the research that we need to look at is very different from other countries. Then, of course, the second one is the innovation. Innovation, what it means is that we should be able to provide the services to the industry. So we will be taking the industry problem statement into the center, and then we will engage our talents from institutes, and then together we'll come up with better solutions so that we can help the companies to move up the value chain, also to address the difficulty that they face in their operations. Now, the third one is enterprise. That is ultimately that we would like to have a launch pad, that kind of environment for the startups or potential spin-off to take off from Singapore. So Enterprise Singapore brought in a lot of the accelerator, and what Mr. Eugene to show you that we have Trendlines, we have U Labs, we have the Hatch Blue. So these are international reputable accelerator. They're actually in Singapore, and then to support the startup and spin-off. And then ultimately, the next one, very important and critical for Singapore, and I believe in some countries as well, is the training. And for Singapore, since we are not in this space since independence, and there's a severe lack of the talents workforce in the aquaculture space. That's why the training has become very important, and then we are embarking on. So if you look at this four-pronged thing, we in AIC, we set up research center, we have the service facility, and then we're launch pad to launch. For example, there are 4 projects that we are launching through our system. That is, first is on oral vaccine, and then point of care diagnostics, rapid DNA diagnostic chips, and also immunity enhancer. So it's just a teaser to show you what we are doing actually at the launch pad. And then, of course, training, we have a very structured and certifiable programs to support the industry. Now I will go into the details to illustrate what we are doing. And for the tech cluster that we grouped what we talents that we have in supporting this industry actually under the so-called genetics, one group. That's for seed productions. Husbandry and health management is under another cluster. Of course, nutrition, feeds, additive, and feed management, one cluster. Now, at the end, at the back of it is the smart technologies, because we really need to engage the smart technologies to enhance our productivities. At the end, the whole objective is to have a super intensive farming system for Singapore. And what we start off is by looking into the existing industry. What can we learn from it? And you guys most probably familiar with the broader industry, and I find that this industry is indeed a very interesting industry that we can actually learn a lot from it. If you look at the performance over the years, how this broader industry has been developed, if you look, it's actually over 100 years, and over time, you can see that the early phase of the development is actually focusing on nutrition, feeds, and husbandry. Right, and the later phase of the development in performance is actually go into better disease and health management and genetic and breeding. So the major contributors to the performance, okay, for the early phase of the development is actually nutrition, and then the later phase is actually genetic and breeding. Now, the whole process in broader industry took 100 years, and now we're starting to have another 100 years to develop a better performance animals for this industry. So we like to compress it. So thanks to the advancement in technology like industrial revolutions, and also the technologies that we are using right now, like they say nutrigenomic, nutrigenetics, and so forth. One of the speakers in session 2 will talk about the genomics technology. So basically we're using all these technologies to compress, okay, the development in the aquaculture industry, improve the performance of the animals in the shortest possible time. And we are learning from the broader industry and how the technology roadmap can be followed through this particular industry. Now, for us to support industry effectively, we need to have infrastructure. So very uniquely for this particular industry, is the infrastructure actually driving the strategy? And for AIC, the main campus we have here is the building that we have, and then we have the facility for us to do a lot of R&D, right? And this is our main buildings where we have RAS system for both fresh and marine species. Of course, we need to have feed mills and live feed culture system for the feed formulations for feed trial. And of course, the testing lab and so forth. And we do have the disease challenge facility, because this is very important. Yeah, because warm water species, they will have a different disease challenges, and we do have a disease challenge facility. And on top of that, we need to have a proper farm, because a lot of the scaling up, we need to go into the farm. So fortunately, we have managed to get a small piece of land that allow us to do a lot of the research and use it for training. Yeah, so this is the layout of the farm where we have actually water coming in for us to tap into seawater, and that's where we have our water supplies for our research and training purpose. So the facility become very critical in supporting this industry. Now, what next? We need to have investigation lab. That's very important that all the biosecurity matters need to be settled for our farm operators. And also in Singapore, we are going for the good aquaculture practice as well. So again, all these certifiable things require testing and compliances. So this will be the investigation lab that we will have the basic standard health packages for the farms, and also we have the service on demand that is based on the needs of the farms, and we were able to go deep into the investigation and to solve the problem for the farm operators. Now, the training become also very important, as we say that we need to have workers that understand the so-called super intensive farming system. It's very different from traditional farming. And we have this formal training program. It's actually 6 months full-time training, because we need to quickly train up the personnel and roll them into the industry and start working. And this is full-time on-the-job training. So we have basically covered what they need to know and then allow them to work in our research farm. At the same time, 6 months later, they will be placed out to the industry. Another training program that we have, we find is very critical, is actually biosecurity. So again, this one we're working with our partners, that is the NTU, School of Chemical and Biomedical Engineering, and also one of the center for the aquaculture research, innovations, and education. So we are actually administering this biosecurity courses so that the farms can be well protected in their operations. Now, another one that is coming up, that is the Singapore Standard for Good Aquaculture Practices. So again, when Singapore government is going to roll out these standards to the farm, and we need to support the farm operator as well, so that their workers is able to comply to the regulatory requirements. So again, we have this type of very powerful certifiable short courses, and make sure that the workers are up to the speed. Now, the next thing we're looking at is the industry revolutions. As we need to compress the development cycle of the species, we certainly cannot have had another 100 years to develop the one species to achieve the performance that we see with the broiler. And we need to bring in technologies from other industries. So industry revolutions, there are a lot of technologies and engineering which can be coming in to support. So we become a conduit now, working with the technology supplier and see how can they help us to move our aquaculture industry to next level in the shortest possible time. So these are some of the typical technologies and engineering that we are looking at, and see how that can be deployed to the farm. Now, ultimately, our model is like this, that it has to be a public-private community partnerships. In other words, it has to be a whole value chain. Now, we start with the first layer, that is we must have investors, someone who is very interested in this industry. In this case, for example, in Singapore, obviously is the Enterprise Singapore, right? And under the Ministry of Trade and Industry. So this ministry will be the one that investing in the industry. Now, the public, that is the talent. So we tap into the talent industry. We cannot allow us to run independently in isolations. The best is coordinating approach. Everybody put their hands on deck together and to support this industry without cannibalizing or create a silo environment. So we brought in the talent industry in as a public sectors. And then, of course, the private will be the one that they have the technologies. Now, the technologies, we don't restrict only to Singapore, like IPI. They have a lot of industry from the regions and from international. So for aquaculture, we are looking at the technologies coming from EU, Japan, and Israel, because they do have a lot of wonderful technologies that we can actually learn from them and see how can we translate those technology into a warm water species or warm water environment, and also in a super intensive farming system. Now, ultimately, it has to go to community. That's important. That's the outcome. So, of course, the first community we're looking at will be in Singapore, but we do look into the neighboring country like ASEAN, and we go as far as India, even to Middle East as well, in this case, Oman. So this is just the so-called the usual community that we will go and engage. And one reason for that is our farm operator, they actually using Singapore as the innovation hub, where they do the innovations and developments. And then when they do the grow out, they actually go to the countries outside Singapore. So we have to follow them. So as a result, they are the community that will be able to benefit from what is developed in Singapore. Now, another system we try to create is actually working with the bank. So for example, Asian Development Banks within ADB, it has its own network, its own country members which they need to support. So we are actually writing on the ADB, their network, where our technologies develop, where the technologies developed in Singapore can be exploited or can be exported to the regions and fast and efficient. So this is the whole modeling that we do, that we have the public-private community partnership, and of course we need a driver. In this case, the driver will be the AIC. So with that, this is my last slide. Basically, we have one identity, that is the AIC. We representing almost everybody in Singapore from the talent industry, and we have one goal, that is to serve the industry. Thank you to you, Dr. Z.
Thank you very much. Thank you very much, Dr. Lee. Appreciate that overview of the translation and support from training all the way to trials and helping company, scale as far as they can go. Now, without further ado, let me have great pleasure to invite our next guest speaker, Dr. Orvin Fleming Jensen, CEO of Nofima. Nofima is a leading institute for applied research within the field of fishery, aquaculture, and food research in Norway. Dr. Jensen will share on frontier developments on aquaculture, genetics, and feed optimization. Dr. Jensen, please.
Thank you very much, and good morning to those of you in Europe. Good afternoon to those of you in the Southeast Asia. It's my pleasure to be here with you today. Thank you very much for the invitation to present some of the views, and it's kind of prolongation from Dr. Lee's presentation. So what I will do is try to focus on two areas where there are great potential for further growth. Nofima is an applied research institute with more than 300 scientists and technical staff, and we have a full infrastructure, revenue about 105 million Singapore dollars, and a surplus of 3 million, which is reinvested into R&D.
Dr. Orvin, will you be sharing your slides?
I think I'm sharing my slides.
Okay.
Sorry.
Yes, it's okay.
I was just, can you see my slide now?
Yes, if you can go into view mode, please.
There?
Yes, please. Carry on. Thank you.
Sorry. Our strategy is fairly simple. It's to enhance the competitiveness of the aquaculture, food, and the fisheries industry. And by doing that, we do research and innovation, which is kind of investing to create results. Then we transform the results into money in the industry. So then you have research knowledge-based innovations in the overall sector. I think if you are looking at dealing in this area, you have to understand the value chain. You have to understand the value chain from the input factors the whole way out to consumers and the markets. And to do that, as my previous speaker was saying, you have to have a state-of-the-art infrastructure, but you also have to have an interdisciplinary approach to your solutions, and you have to find the opportunities. Today I will look at two areas, which is genetic and breeding and feeding aqua feed. But before doing that, in the future you will see there is a convergence of biology and technology, both related to new production systems, to productivity, efficiency, robustness of your farming solutions. Fish welfare and fish health will become more and more important, and also the word sustainability in all three aspects, both environmental sustainable, financially and or economic sustainable and social sustainability. But when we are looking at this interaction between biology and technology, you can't fool biology with technology or money. So you have to have this broad and deep and understanding on what is going on in the value chain when you are trying to find new solutions. If you look at genetic improvement, as was said in the previous, we have done this for years. If you look into traditional plant breeding and agriculture, Mendel and so forth. But over the last 50 years, 70 years, you've seen a tremendous gain in productivity from breeding technologies. Poultry is maybe the one which has the highest growth, but you see in the salmon and the salmon aquaculture, the growth rate is also now improving very well. One of the challenges, and it's a part of it, that too many breeding technologies are not being applied in the aquaculture sector. So if you take a look at what has been going on, that is from conventional selection into marker assisted, into using many gene markers, genomic selection, full genome, GMO, and CRISPR-Cas9. And then, of course, this is possible because we now have a full mapping of the salmon genome. We have a full mapping of husbandry genomes, and the cost of doing this kind of analysis has gone down quite significantly, which makes it possible to apply some of these technologies. However, the increasing toolbox is maybe not just possible. If you look at genomic selection or conventional selection, family breeding, and so forth, there is a proven effect. Unfortunately, it's still an slow uptake of technology. Maybe there are some examples like the gift tilapia project that was started in 1988 in the Philippines and has spread out throughout the world as an excellent example of improved breeding technologies. But in addition to the genetic gain, also reduced cost, improved cost, but 40% of the aquaculture production today is being used with not improved stocks or improved input factors. So gene technology has a great potential by being a part of the focus, and it will be even more powerful in the future. But if you then take the other technologies like full genome selection or GMO and CRISPR, then you have regulatory challenges, you have ethical challenges. Well, we are moving on life. It's a new technology, it's not yet been proven, so the long-term security has not been documented, and consumers might have some concerns that it's not healthy. At Nofima, we are doing CRISPR technology here as an example, that we are using the Pacific salmon, which is resistant to salmon lice, and try to use those genes in Salmo salar, the Atlantic salmon, to see if we can make the Atlantic salmon resistant to salmon lice, which is a key challenge in the salmon farming. But still you need a lot of development. And then the breeding objectives has a lot of potential in new species, domestication, here illustrated by cod, looking at the growth rate, the feed conversion rate, meat quality, disease resistance, and fish welfare and health, and so forth. If we are to change the picture a little bit and take a look at the feed cost is the major cost driver in salmon production or in animal husbandry altogether. Miscellaneous is of course another factor where the cost is increasing quite significantly, and that is related to treatment cost of sea lice and wellboat and other services that we didn't see back in 2000 and in early years. But feed cost is a concern. But taking a look at the feed, there is a major shift because we are at the limit of what the ocean can do when it comes to marine resources, and we are replacing marine resources with plant or vegetable ingredients, and so it's becoming more sustainable in a way that the feed is changing, but you're also using new virgin materials, and you're using byproducts from other animal production as protein sources in a kind of circular economy way. There is a lot of potential for new feed sources, and those are being investigated. Of course, you have controlled production, which is kind of circular, using single cell proteins like yeast, bacteria, microalgae to produce protein. Then you have insects and or side streams from the fisheries or animal husbandry, like feather meal, proteins, and so forth. Or you have marine resources that are not threatened by overfishing or overexploitation, like mesopelagic fish living from at depths between 100 and 1,000 meters, which is not today used, but we are researching on that. You're looking at mussels or filter feeders, or you're looking at kelp and seaweed or microalgae as part of input factors in the protein. However, despite all the search, these things have to be looked into in a more controlled way. In the challenge, you have to have a low CO2 footprint. You have to have a sustainable production that is within the bearing powers of the nature, that you are not destroying the ecosystems. You have to, and you should look at the use for non-human consumption, that if you want to produce, if you can use it, the proteins to feed people directly, you should do so. It should be available in large volumes at low production cost, low processing cost, and nutritional efficiency. And then you have to look at the resources from process feed, the process technology, feed technology, fish nutrition, and then fish to the market and how we eat. And at Nofima we have sorry, it's just telling me that my time is up.
Oh,
it's just we have, and then also an Aquafeed Technology Center at Nofima, where we are working with the industry. So thank you for listening.
Please stay on.
Food for all. Thank you very much.
Please stay on to Dr. Jensen. Thank you very much, and I get Zubi on board as well. Very quickly, in interest of time, I'm going to just put 2 or 3 questions. They are burning questions, and I'll first direct to Dr. Jensen. I think this is a topic we talk about, CRISPR and genetic. So one of the question is, what is the regulation or obstacle for GMO fish, and is there a difference between the treatment with GM or gene edited? So I'd like to hear your view on that.
Well, there are one company, Aqua Bounty, which has made a gene-edited salmon. They have got approval in Canada and the US, and are now in the market introducing the gene-edited salmon. Hasn't received the market attraction that one had would have expected due to a lot of consumer concerns. When it comes to CRISPR, there is still major challenges related to the regulatory side. EU has ruled that it's to be considered as GMO or your genetic modifications according to a ruling back in 2018, and it's very restricted. In the US, they are open to CRISPR, but it's not if it's related to human use, but there's a lot in plants, among other things.
Yes, maybe by extension, how is your suggestions to regulator, even so in this part of Asia? How do they take this perspective on CRISPR and the development in this area?
I think you should have an open mind to the technology. It just received the Nobel Prize, and it's very clear that CRISPR technology is a more precise way of monitoring and doing genetic modifications in the way. And you should have a strict that it's allowed for use in plants and in animals, but you should be very restrictive when it comes to introduction into humans, as we haven't seen the long-term safety part of it yet. But I'm a pro GMO. I'm a pro personally, pro CRISPR, but it has to be developed in a very diligent way with the concern of the consumer. And you should not underestimate the consumer acceptance to these kind of new technologies if you're introducing it into the market.
Understand. Thank you very much. And I think there are a few questions for AIC, but I've just summarized it from a startup who wants to use your facility to do this. Do they see you as a service provider or incubating startup? And particularly are concerned about IP. Yeah, so Chiwi, over to you.
Yeah, our position is we are the service provider, right? We are supporting the startups. Now, of course, a lot of startups, they don't have the means to establish the facility. So we are incubating them so that they can actually use our tanks, our facility for their business development. So for the IP side, so we're very clear that is most startup or most company, they come with IP protected. So that's usually not an issue. It's just they need to validate, they need to improve it further. So if any foreground IP created, it actually belongs to the company. Yeah, the company owns it. So we're really clear we're not interfering with IP. IP is usually resided with the sponsor where they have. And most of the company, they actually comes with the IP well protected. And they engaging us is because they need to validate. Yeah, and a lot of times it could become from investor. Investor want to invest in the company and they need to validate the claim. So they actually engage us. So can you do this for us, right? Or later on help them optimize it to make it even better. So that's a position that we don't do commercial. We stay away from that. And we are here as a government, part of government ecosystem is to support, as many industry or company as much as possible.
Okay. Thank you, Dr. Lee and Dr. Jensen. It's really in view of time, I have to wrap up this panel discussion. It has been very enriching one to understand the support from applied research to translation to support the aquaculture industry in this sector. Now, without further ado, I'll be glad to now hand over the next two sessions to the next moderator, my fellow colleague, Ethan Siu. Ethan, over to you now for the next two sessions. Thank you. Thank you, both keynote speaker.
Thank you very much.
Thank you.
Thank you.
Thank you, Dr. Z. Hello and good day to everyone. My name is Ethan from IPI, and I'll be your moderator for the next two sessions. I'm sure you have drawn meaningful insights into the challenges and opportunities in the aquaculture industry from our keynote speakers. So without further ado, let us dive straight into our first session on feeds and nutrition. The first session will touch on improved fish meal formulations using enzymes and peptide-based technology. Next, we will look at how krill meals enable the production of affordable, safe, and high-quality seafood. I would like to remind all participants that should you have any questions during the presentation, please type them in the Q&A panel at the bottom of your Zoom window. We will address these questions at the end of the session. I would like to introduce our first speaker, Dr. Thomas Wilson, aquacultural nutritionist at DSM Nutritional Products. Dr. Thomas is an aquaculture industrial veteran in Asia with 30 years of experience under his belt in feed meal formulation and operations. Today, Dr. Thomas will be sharing with us on the topic of formulating with enzymes, improving feed performance and sustainability. let us welcome Dr. Thomas.
Okay, is that being shared properly or not yet? Here we go. Okay. There. So I believe you should be able to see my screen. Thank you very much for inviting me to come and talk to you today. I appreciate everyone who's taken the time to come and listen to my talk. So what I'm going to be talking about today is formulating aquafeeds using feed enzymes. And this is for the purpose of improving feed performance as well as making our feeds more sustainable. Now, in addition to being a consultant at DSM, I have long experience in aquafeeds in Thailand, and I've been an actual user of both phytase since about 2006, and also more recently I was using Xylenase. So I have practical experience in this that I want to share with you today. So first of all, enzymes are among, just a second, I'm not seeing the top of my slides because of the overlap of the menu bar. Okay, well, anyway, enzymes are long proven in the livestock and poultry feed industries because these feeds are mainly formulated with plant ingredients. And many of these plant ingredients contain anti-nutritional factors. Probably the three most important are phytic acid, protease inhibitors, and also non-starch polysaccharides, which we could call shortened to NSPs. Now, if you think about NSPs, what we're really talking about, and you're all going to be very familiar with, is dietary fiber. So in the upper right-hand corner, you can see the structure. You can see the structure of a slice of tissue, and you see the cell walls, and inside the cell walls, there are these globules of nutrients. And if we don't break down those cell walls in fish that don't have these kinds of enzymes, then these nutrients won't be available. And in the lower picture, you can see the effect of the plant cell walls, that they're made up of quite complex structures. Now, in the past, we weren't really worried much about using enzymes in aquafeeds, but as we've been reducing fish meal using more and more plant proteins, then this has become now quite of interest and is expanding widely across the globe. So phytase improves mineral nutrition by reducing phytic acid's impact. And in the upper right-hand corner, you can see in that white box, this is the full molecule of phytic acid. And at the very center in the dark circle, you can see is a molecule of inositol, which is actually a vitamin. Now, attached to the inositol ring are 6 negatively charged phosphate groups, and these are not biologically available. They're very strongly bound. So if we look in the center in this bar graph I've put here for you, the full width of the bar graph is the total amount of phosphorates that's available in that ingredient. But looking more closely, you can see that the blue part of the bar is actually the part that is not available because it's bound up in phytic acid. And one of the reasons we want to use phytase enzyme is we want to release that and make all of that available. So if we're looking at the timeline for the process, as you can see over on the right-hand side, that we start out with IP6, and in the acidic stomach, as soon as the phytase enzyme is in there, it starts to chop the phytic acid, phytase, the phosphate groups off one by one. And at some point or another, we will then have this material move into the intestine, which has an alkaline pH. But in here, there are alkaline phosphatases that will chop off the phosphate groups until in the end, we have free inositol and 6 phosphates that can be used for nutrition. So if we want to see the proof of phytase's improvement of mineral and nitrogen retention, the summary, we don't need to deal with what's in the black. Just look at the bottom one, the one that I put out here in red, that phytase improves the skeletal development in feeds, from feeds with low digestible phosphorus and zinc. Now, we may assume that the phosphorus is in the feed. We may assume that the zinc we've added to the feed is all available, but because of the presence of the phytic acid, the phosphate is not available. And what ends up happening is, if you look at the right-hand side, I have two photographs of tilapia and carp. And the upper tilapia is a fish that is being fed with phytase enzyme plus probably a high protein plant diet. And the lower fish is a fish which is being fed a similar diet but without the phytic acid, without the phytase enzyme. And you can see that due to a deficiency of available phosphorus and zinc, that the fish has a shortened skeletal structure, and in some ways we could say that it is stunted. So moving now on to protease. Protease improves protein digestion by enhancing endogenous protein digestion. So, we know that the protein component of our feeds is the most expensive part, and we want to maximize the amino acid digestibility to get the best performance from our feeds. But some of our ingredients contain protease inhibitors, and then also in some cases, due to regulations, we treat our animal protein meals at high temperatures to sterilize them, and this reduces protein digestibility. In the upper right-hand corner, we have this graphic showing, first of all, the trypsin enzyme from the stomach and the intestine, the one that's already there. But the thing is, it's not very specific, and it works fairly slowly. And we also have to worry about those protease inhibitors stopping this enzyme from working. The thing is that when we put protease enzyme into the mixture, the activity is much, much faster. The enzyme is not nearly as specific. It chops the material up into much smaller pieces, much, much faster. So this improves the speed and efficiency of digestion. And it also makes these peptides easier to digest. One curious fact is that protease enzyme actually works better on poorer quality proteins. Okay. Moving now to carbohydrate utilization, Xylenase and a number of other cellulases, they basically make indigestible fiber digestible. And there are two classes of NSPs. One is a water soluble. They gel and they cause a high viscosity in the gut. They affect a gut passage time. They make it difficult for digestive enzymes to find the substrates to digest and make it difficult for nutrients to be absorbed. The second group is the water insoluble. These are basically just large molecules, and the nutrients get trapped inside, and the enzymes just simply cannot find them. Looking at the upper right-hand corner, you can see an intact molecule of a xylan from corn, and the arrows show you where the xylanase chops this molecule up into a bunch of smaller pieces. And so what this does is it makes starch more available, fat more available, amino acids more available. All of this then is available as an energy source in our fish. And probably the most interesting thing is that these short segments of NSPs are basically functional prebiotics. Now, enzymes reduce environmental loading because the more retained nutrients we can keep inside the fish, the less nutrients that are lost into the environment. And of course, with intensive aquaculture, it's very important that we try to keep good water quality. Otherwise, it causes lots of issues with our fish husbandry. Now, worldwide, aquaculture releases about 100,000 metric tons of phosphorus per year, and this is a very significant amount, especially here in Southeast Asia and East Asia. DSM has done some research. This is the data that's being shown in the green box, where when we use 2,000 units of a DSM phytase, which costs approximately $2 in a tilapia diet, we were able to get as much phosphorus back as adding 14 kilograms of monocalcium phosphate to the diet. And at today's prices, that monocalcium phosphate would cost approximately $8.40. So you see, it's nearly much, much cheaper to use phytase enzyme. Now, in the upper right-hand graph, this is Clarias carapinus, walking catfish. And this data shows that when we add phytase enzyme, we see a significant reduction in the amount of phosphorus excreted by the fish. And in the lower graphic, this is showing the improvement in protein retention by the use of Xylenase enzyme. So we're actually measuring protein efficiency ratio. And the curious thing is, in our diet with the 0 fish meal and the highest amount of soybean meal, we actually see that the protein retention is higher than even our much better fish meal containing diet. So in conclusion, if we want to consider the benefits of adding our enzymes to sustainable feeds or highly plant-based aquafeeds, we can use phytase, protease, xylenase, or a combination of all of them or all of them together in a cocktail. We get a much better retention of our nutrients. So of course, we get better feed conversion ratio, better growth, we get a cleaner environment, and we also are able to save costs because this allows us to use more efficiently the nutrients that are already there, leading to a better total sustainability of our feeds. And in my opinion, using enzymes in feeds, especially those feeds high in plant-based ingredients, for me, I consider enzymes to be an essential component, not an optional component anymore. Thank you very much. So that's the end of my talk. Thank you very much.
Thank you, Dr. Thomas. Looks like we should start supplementing some of the fish diet with the enzymes. Next, I would like to introduce our second speaker, Dr. Nisim Khin, founder and CEO of Aquinovo. Dr. Nisim has over 20 years of experience in technology commercialization and business development in the biotech and agriculture industry. Dr. Nisim will be giving a presentation titled Peptide-Based Feed Additives for Farm Fish. Let us find out more. Dr. Nisim, please.
Hi, everyone. Thanks, Ethan. Thank you, everyone, for the invitation to speak here today. Let me just share the screen. Okay, I'm sorry. So we are going to talk a bit today about the peptide-based feed additives. And let me maybe start with a brief introduction of where we are and what we do. Aquinovo develops innovative peptide feed additives that generate more fish with less feed. Obviously, that addresses one of the key challenges of aquaculture, which is how to sustainably increase fish yield with less feed, less input, no change in practice and infrastructure, and so on. And the outcome of that is, of course, increase in fish farm profit. Now, beyond that, beyond the increase to the farmers, we are promoting, by doing that, we are promoting important SDGs like preserving life under the water, food security, poverty and hunger, since we have more profit. And while using less feed, we can actually reduce the GHG emissions from that feed, which is the main component of the aquaculture GHG emissions, and therefore contribute to climate action. And when synthesizing our peptide, we're also relating to responsible consumption and production. So that is where we come from and what we do. And sorry, okay, and okay, so if we look more into the issue of feed and aquafeed, we know that aquafeed is one of the, of course, major important parameters in the quality of of fish and the yield of the fish. And feed additives is one of the components that brings innovation into the feed. And when we look on feed additives, there are, I would say, two distinctive groups. One relates to the feed quality, binders, emulsifiers, and so on. The others have more nutritional factor, mainly free amino acids, but also vitamins, minerals, etc. Alternative proteins also come into this category as they replace other proteins, nutritional proteins. And the third one, the functional additives, include all those type of additives that have an impact on specific parameters such as growth, gut health, feed efficiency, general health, immune system, and so on. And they have various materials in those groups, starting from plants, yeast, and animal extracts, going all the way to mycotoxin binders. And when you look on which of those functional additives actually include peptide, that would be mainly the extracts, and in some cases hormones that are used mostly in tilapia for sex conversion, or in other industries as growth hormone. But those are the main places where you can actually find some peptides. And the question is, maybe before we go more into depth into why we need to look at it, is why do we look at peptide at all? Why should peptide-based feed additives be a target? Well, as a quick reminder, peptides are short polymers of amino acids. And when you look on peptides, they have some powerful features that are enabling use and are being used in other industries, such as the cosmetic and pharma industry. They exist across all organisms and body systems, but at the same time, they are very specific. They bind to specific receptor, and they have very conserved sequences between species and among species. At the same time, there is a variation due to various mutations that change only one amino acid in the sequence, and this allows flexibility and adaptation when you want to use peptides on different receptors for other targets. Peptides are also biodegradable, and it could be a double-edged sword, because you want it to be stable enough to be orally taken and do what it needs to do, but at the same time eventually be cleared and degraded from the original form. So we have a lot of data on peptides. As I said, it's very common in other industries, and so that you can use in order to try to exploit all those important powers that they are giving us. At the same time, we have some serious limitation, which is the production. Commercial production is the limitation. When you are talking about scalability to hundreds of kilograms of tons, it became to be a very complex issue, and the cost is going up rapidly. So this is the issue of why peptides, and going back to the going back to the source of peptides, current source of peptides in feed additives, it's mainly extracts of plant, yeast, and animals that are generating bioactive peptides, along with a lot of other peptides that are mostly nutritional and don't have a specific active known role. And those bioactive peptides usually work on two components: the animal physiology, or the microbiome, or sometimes even both. And when we're talking about physiology, the main targets are reproduction, gut physiology, and immune system. Eventually, it all leads to the same impact we are looking for. They are all impacting growth, feed efficiency, survival when we have challenges, like let's say we are using antimicrobial peptides. And again, nutrition, where the peptides are being degraded, or there are a lot of unknown, inactive peptides that are in there and are used as basically efficient vehicle of amino acids. So this approach is having some advantages and disadvantages. I mean, the first one is very obvious, because when you do extracts of plants and animals, you use byproducts of meat and fish industries that was estimated to be around 60 million tons a year, and also use bycatch in the fishery industry. It's a source for a vast diversity of peptides, both known and probably a big section that is unknown. And it can, again, serve as active role doing something, but also may have a nutritional effect for just carry on the amino acids. But at the same time, there are serious limitations. Production at scale is a problem when you are talking about a heterogeneous raw material. And the same goes to the most limiting factor, which is batch-to-batch consistency. Heterogeneous sources, both in terms of species and places where it was done, makes a real problem going into specific peptide that you want to achieve. Limited data is known about the unknown active ingredients, so there are also issues of safety and efficacy. There is low palatability known for some of those peptides, and there are a lot of question marks that needs to be answered with respect to stability to extrusion. When you're talking about plants, there are a lot of anti-nutritional factors that needs to be taken into account. And when you go more higher into sustainability discussion, you have to look on eventually the land use and land use change for growing those plants. When you're using fish hydrolysates, you're of course using waste, but you're not reducing bycatch. So that's another limitation. So, sorry. Overall, we have, of course, advantages and disadvantages. And what we in Aquinovo are doing, we are looking more into the, I would say, more rational approach, more targeted approach. We are talking about a targeted bioactive peptides that is basically the same thing that have been done for years in the pharma industry, looking on what are the endogenous peptides that we know about, what are the roles, what are the receptors that are being used, and then looking for what do we want to do in order to make this process more efficient or giving us the beneficent results we want to have. So for example, if you're looking on gut physiology, we're looking on endogenous peptides in the gut, on their receptors, on what type of processes they are involved in, for example, gut motility, digestion in the epithelium, immune system, and so on. And then we design and chemically synthesize those peptides. And we have a family of peptides that are non-hormonal, of course, bioactive, short peptides that are targeting specific receptors in the GI tract. And we have shown in tilapia that there is an improvement of both growth and FCR. And this is very important because the targeted approach allows you to bring in peptides that you have designed it to a specific receptor and characterize their role. And of course, you need to synthesize them, and that's another important issue that has to be taken into account. Now, here is an example of one of our large-scale studies performed in Asia with red tilapia. To the left-hand side, you see the control group. This experiment was done with mixed population of both male and female of tilapia that received our peptide in their feed for about 4 months. Sorry, and then continued to be grown on regular feed for another 6 months until harvest, reaching the final commercial weight. And what you can see clearly is the differences between those two peptides in terms of weight. And we have demonstrated in the data that we have a significant increase in average weight and a significant improvement in FCR at the end of the period of the whole experiment of the 10 months period. We also saw that we can see a different distribution of the feed that will help farmers in grading. We see more big fish and less small fish. That helps not only in grading, but also in revenues, when big fish usually have higher price per kilogram. We see no effect on fish health and no accumulation of fish in the water, no accumulation of peptide, in the fish and in the water. In terms of the farm, it can be translated very easily to high profit. And again, in terms of the global effect, we are having more fish promoting those SDGs that we have discussed earlier. So to wrap it up, we are talking about, let's remember eventually what the target is the target is to feed the world population that is going to be about 10 billion in 2050. So in order to do that, we have to harness the biotech tools that we have, and peptide is one of them. And we can do it by generating innovative peptide-based feed additives that will target specific receptors and therefore can improve growth and FCR. In simple words, generating more fish with less feed and contributing to other sustainability issues. And this is my my input for today. Thank you very much.
Thank you, Dr. Nisim, for your sharing and a crash course on fish meal replacement. Next, I would like to invite Dr. Tibiabin Benitez Santana, R&D Director of Fish Nutrition and Technical Aquaculture at Aqua Biomarine, to share about krill meal as a sustainable ingredient for fish feed. Dr. Tibi is a fish nutritionist with over 15 years of experience in the aquaculture industry, focusing on understanding fish nutrition, feeding, and health. Dr. Tibi, please.
Thank you so much, Ethan, and thank you so much to the organizers for giving me the opportunity to be here today and talk about krill. During the last years, the aquaculture industry has been looking for alternative sources to fish meal and fish oil. Therefore, it is crucial to find sustainable raw materials with a good nutrient profile in order to improve the performance in aquafeed. The aquaculture industry is aiming to use 6% of these novel raw materials into the formulation by next year. And Aqua Biomarine believes that krill can play an important role in these novel raw materials. But first, let me introduce you what is krill. Krill are small crustaceans and they can live in all the world's oceans. So outside in Singapore, you have krill, but it's only in the icy cold waters of the Southern Ocean in the Antarctica where they can be harvested from the wild. Krill plays an important role in the trophic level connection near the bottom of the food chain, and their biomass is one of the largest in the whole planet. The Antarctic krill fishery is one of the most sustainable in the world due to the near zero bycatches and fully transparent operation. And here you have a picture of the Antarctica, where in this area called Area 48 is the only place where the krill can be harvested. Last year, the Antarctic krill fishery received the A rating by the Sustainable Fishery Partnership as the only reduction fishery with a biomass considered in a very good condition. From this krill, we obtain the krill meal, which is rich in proteins, omega-3 phospholipids, and astaxanthin, and is intended to partially replace fish meal in fish diets and also in shrimp diets, and also plays an important role in plant-based diets as well. The krill is alive, so depending on the season, it might be fed with red algae or green algae, and therefore the fat content might be higher or lower. But what we know is that from this fat content, 40% of the fat is packaged into phospholipids, and this is very important for early stages in marine fish overall. Also contains the important omega-3 fatty acids such as EPA and DHA. So the difference between phospholipids and triglycerides is that the phospholipids can be used by the membrane cells in a better efficiency way. However, the triglycerides, which are a great source of energy, has to go through the liver in order to be converted into phospholipids. So only a little part is going to be used by the membrane cells. So this is the major difference. But I like to think about krill not as a raw material. I prefer to define it as a beneficial package of different nutrients, because besides of having these important omega-3 phospholipids, also contains astaxanthin esters that not only gives the pinky coloration in the skin of the fish or the salmon fillet, for example, also contains antioxidative properties that are going to protect the meal, the feed, and also the tissues of the animal. Contains chitin coming from the exoskeleton of the animal, which seems that also has immunostimulatory properties in order to boost the health of the animal. Contains cholesterol, taurine, vitamins A, D, and E. But the beauty of the krill meal is that also acts as a feed attractant, because we know when we reduce fish meal, we can reduce the palatability. So that's why it's important to add something that is going to attract the fish, because we know that the fish are very picky, and overall in stressful periods, they might reduce the feed intake. So thanks to the nucleotides, water-soluble amino acids, and TMAO, these compounds are going to keep the feed intake up and running. Aker BioMarine is a company driven by science, so all the claims that we have about Krill Meal in the fish performance, quality, and health have been proven from the scientific point of view. Why these parameters? Because we know, for example, for the farmers, they really want their fish to grow as fast as possible in order to reach the harvest size. Quality, this concerns all of us because we are visual consumers. We want to go to the supermarket and buy a good fish with a good shape and also a good fillet as well. And robustness, this is related to the health of the animal. So animals or fish with a good, healthy liver, intestine, heart, and other organs, it's gonna facilitate the challenges that the animals might need to face during the production. And now I'm gonna give you some examples about the effect of Krill Meal in performance. So here you have different examples in early stages for sea bream, tilapia, olive flounder, and European sea bass. And we have seen that reducing fish meal and adding Krill Meal, it has a positive effects in the final body weight, in the performance. And this is nicely correlated with the decrease of the feed conversion ratio. So the animals are using the nutrients in order to grow. And as here you can see that in the tilapia study, only with 1.5% of Krill Meal inclusion levels in the feed was enough in order to boost this growth. So not because you add more krill, you're gonna get more benefit. It's about playing with the inclusion, with the nutritional requirements of the animal. Also, this study was conducted in South Korea, and we found that decreasing by half the fish meal inclusion level and adding 6% of krill meal was significantly enough in order to improve as well the final body weight. And in the European sea bass, the same. Only 5% of inclusion of krill meal, we could see 20% more of the growth. And this is because the animals really like what they eat, and this is due to the compounds that increases the feed intake. When it comes to the fillet quality, we have seen a very positive result in the salmon fillet quality. When it comes to the firmness, as here you can see in the first graph, it was increased by the addition of krill meal into the salmon feed, and it's because the omega-3 phospholipids are promoting the muscle growth. Also, due to the natural astaxanthin, we have seen that adding krill meal into the formulation, the fillet gets the nice coloration, but not only that, this coloration is even in the whole fillet, which is very important for us as a final consumers. Always, we have seen a reduction of the gaping. Gaping, as you can see here in the picture, is these holes that happens because the animal overall at the end of the summer grow very quickly, very fast, producing this gaping. And this as well together with the coloration, we as a final consumers, we don't want to have this in our fillet. So last year, together with Nofima, actually, we published a new study where actually we could understand why when adding krill meal will reduce this gaping. And it's because a mix of nutrients such as the omega-3 phospholipids, astaxanthins, and maybe others are improving the collagen formation. So here you can see that the fibers are more organized, and that's why there is less gaping in the fillet. And when it comes to the health of the animal, we have performed different studies, for example, in early stages, in order to see if there is an increase of demineralization thanks to the krill phospholipids. And this is very important because it can reduce the skeletal anomalies in later stages. Also, when testing new raw materials, it's very important to test the digestibility. And here in this study conducted in Japan with Seriola, we found that the digestibility was increased. Also, something very important that we have seen in marine fish is that there is a reduction of the fat accumulation in the liver. We have seen that the omega-3 phospholipids mobilize the fat to the different organs where the fat needs to be used. And this is going to make the fish more robust in order to cope with the coming challenges. And also we have seen positive results when it comes to challenger disease. And this study with olive flounder in Korea, we found that adding 6-9% of krill meal was enough to boost the immune system and therefore to increase the survivals. So here you can see some of the main results obtained when adding krill meal into the formulation. Overall, in early stages, we have seen a better quality of the larvae, and also increasing the disease resistance, and also in the finishing diets and improvement of the fillet quality. And we believe that sustainable marine ingredients such as krill meal can lead to sustainable aquaculture. And this is very important in order to provide healthy food for the growing population. Aker BioMarine is a company that is vertically integrated, so we control all the supply chain from the krill harvesting to the sales and marketing. And all of my colleagues have made an effort in order to make innovation, our science department, very good in order to find the claims to convince others about the benefits of the krill. And with this, I would like to thank all of you, and I'm happy to take any questions if you might have. Thank you so much.
Thank you, Dr. TB. I would like to request for Dr. TB to stay on screen while we invite Dr. Nissim and Dr. Thomas to join us for a short Q&A. So in the interest of time, I'll ask 3 questions to Dr. Thomas, Dr. Nissim, and Dr. TB each. So the first question to Dr. Thomas, can we use Aquafeed for all types of fish or shrimp? And how would the use of the enzymes change the quality and taste of the fish meat?
Well, really for fish that we're growing, our biggest impediment is more the fact that for many of these species, we just don't know the nutritional requirements. The research has never been done, or it's very spotty and piecemeal. So this is really a bigger issue, when we're trying to use enzymes. Well, have we provided enough of the essential nutrients that we need in the first place? Enzymes can certainly help you if you want to use plant proteins. The thing is, fish feed is quite complex, and flavors and textures are also related to protein quality. They're related maybe to the amount of vitamins in the feed, and also things like fatty acid profile. So there's a lot of factors that affect the quality and taste of fish, and enzymes could help with that a little bit, but there are other ways that we could manage that better than just using enzymes.
Okay, thank you very much. For Dr. Nissim, what are the views of the use of probiotics or prebiotics in fish feed?
Well, I think fish, pre and probiotics are already being used quite extensively. And I think that it would probably continue to be used. And I think innovation will go into looking for more either, I would say genetically modified, but genetically improved strains that would put those in use in the future.
Thank you very much. And the last question for Dr. TB from the audience. How can consumers be assured of consumer safety and regulation compliance when they use Aker BioMarine's krill feed?
Yes, so we have our departments of quality control where we checked any problems that might happen during the, not only the harvesting, also the packaging, and checking the nutrient composition of the krill. So there is a lot of safety logistics around krill meal, so it's completely safe. And also we follow the regulations of the different markets when it comes to different nutrients in order to be all aligned. So it's totally safe. Thank you.
Thank you very much. On behalf of the organizing committee, we would like to thank Dr. Thomas, Dr. Nissim, and Dr. TB for sharing on how we can produce affordable, safe, and high-quality seafood through feed optimization and nutrition. Now we'll move on to the second session. So the second and the last session of the Aquaculture Innovation Webinar will spotlight the availability of pharmaceuticals for seafood farming in Southeast Asia with different technologies for optimal shrimp breeding. Let us begin session 2 on health and genetics. Once again, I would like to remind all participants that should you have any questions during the presentation, please type them in the Q&A panel at the bottom of your Zoom window. We will address these questions at the end of the session. I would like to first welcome our first speaker, Tara Tingbo, Head of Commercial Development for Tilapia in Asia Pacific and Latin America at Pharmac. Tara is a passionate fish health biologist with 14 years of experience in various species of fish, including salmon and tilapia. Today, Tara will be presenting on the topic of the role of preventive fish health management in sustainable aquaculture production. Let us welcome Tara, please.
Thank you, Ethan. See here. Hello, everyone, and thank you for the introduction and for the invitation to speak. Today, I'm going to talk about the role of preventative fish health management in sustainable aquaculture production. Just 1 second. First, some information on who we are and what we do. Pharmac is part of Zoetis, the world's largest animal health company. Our specialty is fish vaccines. We have 375 employees globally and more than 30 years of experience in R&D on fish health and development of vaccines. Our current portfolio consists of more than 35 vaccines covering 8 different species of fish. The majority of products are for salmonids, but we also have vaccines for sea bass and sea bream in the Mediterranean, pangasius in Vietnam, and tilapia in Latin America and Asia. Globally, 7 out of 10 salmons are vaccinated with a Pharmac product. Pharmac has 2 subsidiaries, Pharmac Analytic and Pharmac FishTech. Analytic specializes in research on fish disease, diagnostics, consulting, and innovative fish health solutions. FishTech develops and markets equipment for manual and automatic vaccination and also offers vaccination services in Norway. Experience show that high quality administration is as important as the vaccine itself. So our mission is to detect, predict, solve, and to prevent diseases for the global aquaculture industry. Many of you have probably seen this slide several times before, but as the saying goes, one cannot get enough of a good story. It shows the use of antibiotics in kilos, which is the green bars, versus production of salmonids in metric tons, that's the blue area, from the mid-80s and until 2019. The first vaccine was launched in 86, 87, and more vaccines were introduced successively after that. In 87, antibiotic use was around 50 tons in Norway, while in the mid-90s it had been reduced to only a few hundred kilos. Notice also the growth in production from around 50,000 tons to more than 1.4 million tons today. And this would not have been possible without vaccines, but several other factors have, of course, played important roles in the evolution. First of all, salmon is a premium product with a production cycle well suited for vaccination. The fish are vaccinated in facilities on land before transfer to sea. Production is industrialized. There are multinational players and a high degree of consolidation and vertical integration. Important work has been done on breeding, nutrition, husbandry, and fish health management, and high-quality diagnostic services have been available. There has been an efficient and successful regulation of the industry, and also extensive collaboration between legislators, farmers, veterinarians, academia, and the animal health industry. Authorities have had a pragmatic approach to registration of fish vaccines and therapeutics, helping to make these quickly available. And last but not least, high margins have of course driven innovation and investment in the new technologies. So what about other species? Big volumes of fish are produced in Asia, but large parts of the production is fragmented and primitive and done on species such as carp, catfish, and tilapia. Farming environments are often challenging, husbandry needs improvement, and fish are harvested at small sizes and sold to local markets at a low price. When intensive monoculture evolves into an industrialized production, which you can see in the lower part of this figure, introduction of vaccines become more feasible. Infrastructure and the required tools and procedures for husbandry and fish health management are usually in place, and profit margins allow farmers to invest in new technologies. Today, industrialized aquaculture of fish in Southeast Asia can be divided into high volume production of freshwater species such as tilapia and pangasius, and low volume production of high value marine species such as groupers and barramundi. And this is where vaccines become relevant. Among the challenges we face when we introduce vaccines in emerging markets is suboptimal husbandry and fish health management. Sometimes we can see that basic improvements are required before investment in vaccines makes sense at all. The disease situation is sometimes complex and challenge pressures are high, and more than one vaccine may be required to make a significant difference. A thorough diagnostic survey is very often necessary. The concept of paying for a solution before you have a problem is new in many cases, and also the ability to invest in new technologies may be low. Authorities in some countries have limited experience with licensing of fish vaccines, and regulatory processes may be cumbersome and unpredictable, which adds risk to the vaccine projects. Furthermore, also restrictions on export and import of biological materials and vaccines for R&D purposes can tend to slow things down. And important for success, sorry, I'll just move this back. Important for success is knowing the species, the farming practice and the industry, and having a proper understanding of the local epidemiology. Establishing criteria for successful vaccination is also key. For example, health checks, vaccination procedures, equipment, and quality control. And we also always seek a close collaboration with authorities, fish farmers, and other stakeholders. And as shown in the salmon success story, a long-term commitment is required. The first vaccine is just the start of a longer journey. And to wrap it up, a shift from therapeutic treatment to prevention of disease is required to facilitate a healthy growth. Antimicrobial resistance is posing a significant and increasing threat to animal and human health, and something needs to be done. Also, good farming practice, fish health management tools, and efficient regulations are required to support and promote sustainable production. Collaboration between authorities, farmers, academia, and the supply industry is key in order to keep improving fish health management. Consumer awareness on animal welfare, food safety, and sustainability is growing, and the reputation of the industry is really important for confidence in the products. And last but not least, everyone should be entitled to aquaculture products that are farmed in a safe and sustainable way. So that's it from me. And if anyone has questions later on or would like to connect, please feel free to contact me on the email address on this slide. And thank you very much.
Thank you, Tarja. Fishes, very much like us humans, need vaccines to stay healthy. Next, I would like to introduce our second speaker, Dr. Marcela Salazar, Scientific Director at Benchmark Genetics, Colombia. Dr. Marcela has been studying the genetics of immune response against viral and bacterial infections for more than 20 years, and has published several articles in peer-reviewed journals, ranging from venomous shrimp to human diseases and immunogenetics. Dr. Marcela will be providing her thoughts on breeding for disease resistance in shrimp using mass selection to genomic tools. Let's find out more. Dr. Marcela, please.
Thank you very much, Ethan, and thank you very much for the invitation. I hope this talk is perfect for the pandemic time, so most of you will be familiar with all the terms and things that I'm going to talk about. In the shrimp world, we have had more than 5 pandemics in the last 23 years. We started in 1990, and the last one was in 2013 with apins, which was a catastrophic pandemic. And we have now always new viruses and new bacterial diseases. So one of the problems is how to handle that in the shrimp world. Well, If we compare our pandemic times now with the shrimp world, we don't have vaccines.
Dr. Marcella, would you mind
Yes?
going to full screen?
I'm sorry. If we compare that in the pandemic tools in the world of shrimps, we don't have a lot of tools that we have now for our pandemic. We don't have vaccines, we don't have therapeutics, but do we have surveillance, which is key in the prevention and treatment of pandemics? We have management, as we can start using decreased densities, which I call the distancing, but we have some tools that we don't have in the human world, as selective breeding, for example, which I'm going to talk about. We will have probably gene editing, as it was mentioned before, but for now, what we have best is surveillance, management, and selective breeding. For selective breeding, we can start from the first that was tried, which is the mass selection. In the mass selection, what we do is infect a lot of animals, either by normal infection in ponds or by control challenge test, and then reproduce the survivals. It's been used largely for the selection of TSV and white spot in Latin America, and it has very good results in terms of survival. Then we can move on to more complex systems as family-based selection for SIP test traits or family within family selection, where we have a lot of animals from different families, from 1 to 200 families and 20 to 50 animals, for example, and we analyze each animal for growth and for other traits such as disease or carcass traits, for example, and then we analyze all the performance, not only of the individual animals, but also on the families. And with that, we can calculate the breeding values for the families and for the individuals. When we do disease resistance, we cannot use the within family component. We only use the full SIPs as breeders. So we are playing only with the 50% of the genetic variability. We can move further ahead and try to understand why some families are better than others, and look at the traits and the markers that are responsible for the survival of the individual or all the families. For that, we take a lot of phenotypes, a lot of fish or shrimps, and we try to translate that into genotypes by doing genotyping with different density chips. The ideal situation is when we can find a gene that is associated with the survival or with the mortality, or if we can find markers that are associated with the gene by linkage disequilibrium. Sometimes we are so lucky that we can find a QTL that explains a lot of the genetic variants, or sometimes we are not as lucky and we have to move further into our other genomics, which is genomic selection, trying to look at all the most or the larger number of positions that we can do in the genome and see what is the importance of each position and each SNP on the final survival or on the final mortality of the animal. That's been described for all species. Holstein, for example, Holstein cows are used as a model because they increase the production of milk and by 20 or 30% just by using genomic selection. As it was mentioned prior, it's been used also on chickens and other species with very good results. Why? Because instead of looking at the animals, we'll be looking at what is that makes that animal resistant or susceptible. When we do the normal selection or family and within family selection, we'll be looking only at the best families, individuals, and we'll be selecting individuals only from the best families, because we don't have the possibility of looking at animals from other families that are not so good, but in which the animals can be very, very good. So if we use only family and within family selection, we'll be using only 5 or 10 families. While if we do our genomic analysis and can select the animals that are very good based on the genes, we can select many more families, increasing the selection differential as well as also decreasing in breeding because we'll be using a lot of extra animals. In terms of selection accuracy and biosecurity, the systems that I describe you are different. For example, in terms of biosecurity, mass selection is not allowed. In mass selection, we'll be infecting animals, and those animals can be transmitting or can be infected with the disease. Those, the progeny cannot be used safely. The family and genomic selection are much more safer for that because we won't be infecting any animals, and they can be considered SPFs. In terms of selection accuracy, on the other hand, the best would be to be able to do mass selection. Why? Because we'll be having not only the selection of the real survivor with the genome and everything, but we'll be also be able to use the immune priming effect, that it's very important also, and it's been described in shrimps and in other insects. In genomic selection, we can have up to 100% of accuracy, depending on what we have in terms of the genes that we identify, and if we have very good markers. And in family selection, we'll be playing maximum with a 50% of the genetic gain when we talk about full sieve families. Now let me tell you what we do in Colombia and what we have been doing in the past 20 years through at least 2 pandemics that I've been working with, the TSV and the white spot. In Colombia, we have 2 different breeding programs for shrimp, for Lithopeneus banamei, one in the Atlantic coast, which is our breeding nucleus, family and within family selection, and it's a biosecure SPF nucleus located in Cartagena, here in the Atlantic coast, and our Pacific program, which we call a mass selection program for white spot. Colombia is the only country in South America with 2 different coasts in the Atlantic and Pacific Ocean, and it's like having 2 different countries. It's 2 programs are separated by more than 2,000 kilometers and a lot of mountains that you can see. The first pandemic that we had to deal with in Colombia was the Taura syndrome virus, which came in 1993. That was the perfect example of the use of mass selection. At the beginning of the pandemic, the producers decided to use the survivors as breeders. And with that, they increased the survival of the animals by 15% in the next generation, each generation. With that success, the Colombian sector decided to start a family selected program in 1997 using the best strategies available from salmon at the time, which was family and within family selection, and decided to work on the challenge test with a heritability of 0.19. And as a consequence, after 2000, all animals in Colombia are now, most animals in the world are resistant to Taura. That is the success story of how to use genetics for controlling diseases. Then in the year 1999, we had another virus, which was white spot. And the best definition for that virus and for our SARS-CoV-2 is a piece of bad news wrapped up in a protein code. That definition is by Sir Peter Medauer, Nobel Prize in Immunology. And I think it really defines what a virus is. A white spot hit Colombia in 1999. We started with the family selection program in between 2000 and 2004, believing that what we had learned with TSV was going to be completely applicable and very easy to solve. While white spot proved us to be a very more difficult virus than TSV, and we had to switch back to mass selection. If you can see, with the TSV, we started from mass selection and went to family selection, and with white spot, we went from family selection and back to mass selection. Why we had to do that? Because in white spot, when we did the challenge test, we had 2 serious problems. One is that was a huge negative correlation between the resistance of the animal, of the families, and the growth of the same families, which was a problem for the sector. And because most of the families, when we did challenge tests, had 0% survival, and in some of them we had only 1 or 2 survival animals. With that and the heritability was below 0.05, so it was very difficult to achieve genetic gains. With that in mind, we decided to do a change of a strategy and go back to mass selection. At that point, we decided to infect millions of animals at PL stage 40, so we could have a lot of animals. We included a lot of different origins, and in the first challenge, our survival was pretty low. It was almost 1 in every 100,000 animals. So with that in mind, we decided to reproduce the small number of survivors that we had. We even had names for them. They were so little, so few. And we found that just by doing that in the first generation of selection, we found almost a 50% survival on the selected lines versus a 2% survival of the non-selected lines. We proved that in Colombia and in challenge test, also in the University of Arizona. With that in mind, we had 2 lines, which was the white spot resistant line, mass selected, and the susceptible line in Cartagena that had been selected for growth. And we decided to see, okay, what happens if we cross them? What will be the performance of the hybrid? And incredibly, we found almost a exact middle survival of the hybrid of the 2. So our next step was, okay, what is the safest way to introduce those resistant genes into our SPF populations and be able to select for an animal that is SPF and SPR for white spot? For that, we decided to use genomic selection and genomic tools and use our pure resistant line crossed with the Cartagena line or with the susceptible line and do a training population for white spot challenge test using a SNP chip of 35K, and also keep some breeding candidates for selecting the best white spot white spot families or individuals to be used as resistance. For that, we did a challenge test. In the training population, we genotype all animals. We measure every animal that died at what time, what day. We took a tissue sample and we sent them to be genotyped. And then from the breeding candidates, we also decided to test our hypothesis if the selection of using the genomic parameters was going to work. So we did a genomic selection proof challenge test, and we also did the dissemination, used the animals for our dissemination line. So the white spot resistant line could be available for dissemination. And when we did our challenge test, the first thing that we looked was at the plots of the genotypes. Were our animals from different populations? Could we distinguish that? And we found that we had our Tumaco population, which is the resistant line, was very different from the Cartagena line, which is the susceptible line. And in the middle, we had our hybrids. We also look at the GWAS results. We were not so lucky as to find a QTL or an important market, but we found polygenic inheritance for both for time to death and for final survivals. And we could have many QTLs. So we had to go for GWAS status. Our evaluation for testing that was to select the parents based on the EBV, genomic EBV estimated from challenge test of SIPs, as I told you. And to try to prove our hypothesis, we designed 3 groups. Those animals that we consider had very good GEBVs, those animals that had bad EBVs, and group, it's not completely random since we have taken out the high and low animals. So we ended up with 3 groups, ones that we thought were performed very good, ones that we knew were performed very bad, and ones in the middle. Then we did a random mating within each group, avoiding full or half-shift matings based on the genomic relationships acquired with the chip analysis. And we produced 60 families, 32 good, 20 random, and 8 bad. When we did the re-challenge test again, we found that the survival of the families that we thought were going to be good was above 50%, and the survival of the families that we knew were going to be bad were around 26%. So we got a 14% increase in genetic gain in only one generation by doing that. What is the importance of doing this? That we were able to predict the behavior of the animals in terms of white spot resistance just by looking at the genome, the genomic characteristics of the parents, and we didn't have to infect the animals again. So this gives us the possibility of doing white spot selection in our breeding nucleus with safe SPF populations without having to reinfect animals or to compromise the integrity and the biosecurity of the breeding nucleus. We were able to do this because we had acquired all the experience that Benchmark Genetic has had in Norway with the genomics in salmon for many diseases. So the technology transfer works from salmon to shrimp to tilapia and to any other farm major species that we can do. And as we have seen in this very interesting webinar, there are a lot of things that we can share between species and that we can learn from other species. And that shows us that collaboration is key in innovation and technology transfer. Thank you. And if you have any questions, I'm really happy to answer them. Also, here is my email in case somebody wants to write direct. Thank you very much.
Thank you, Dr. Marcela, for sharing about your journey to build up disease resistance in shrimp. Next, I would like to invite Shai Ufes, co-founder and CEO of Aqua Therapeutics, to share about the topic of orally administered health management platforms for shrimp. Shai is a molecular biologist and entrepreneur with over 15 years of experience in biotechnology. Shai, please.
Thank you very much, Ethan. And thank you very much for the organizer for inviting me. Hi, everyone. I'm Shai Ufaz, co-founder and CEO of Aqua, a biotechnology company that has developed an oral delivery platform for preventing aquaculture disease. Aquaculture has become the world's fastest growing food production sector and is now widely recognized as an important segment in our global food supply system. Aquaculture is still facing a range of sustainability challenges. Among the more pressing issues are land use and pollution, unreliable farming practices, and disease. Disease is still the major challenge for sustainable aquaculture, causing yield loss, feed waste, and losses of billions of dollars annually. The Aqua approach to facing the disease challenge and to actively support sustainable aquaculture is through RNA-based solutions. Why RNA? RNA has great advantages. It is naturally existing in all living cells. It is recognized as GRAS material by the FDA. And in contrast to antibiotics, it is a precise and targeted solution, an important environmental advantage. Another advantage of the RNA is that no genetic modifications are required for its effect to occur. It can be used as health enhancement material, antiviral treatment through activation of the RNAi mechanism, and as a vaccine, as we all recognize this year with the COVID-19 new mRNA-based vaccination approach. These characters makes it an exceptional material for solutions for the animal health sector. Although many technological achievements have been made in the RNA world recently, RNA delivery, or in other words, how to effectively deliver the RNA to the animal cells, is still the major challenge in the area of RNA-based therapeutics, especially in aquaculture. The Aqua developed an oral delivery platform tailored for aquaculture and specifically for the shrimp industry. It includes the selection and design of the relevant RNA sequence, encapsulation of the RNA using innovative carrier technology, followed by a proprietary process that allows the incorporation of the product into the shrimp feed. The Aqua formulation-based solution allows the RNA to survive the aquatic environment, the shrimp digestive system, and to be absorbed into the shrimp cells, where it triggers a cell mechanism that protects the shrimp from the virus. The final product is a shrimp feed reaching the market through existing distribution channels. The Aqua's first target product is a solution for the white spot syndrome. We just heard about it, and so you all know that the white spot syndrome is an example of a globally spread viral disease that continues to spread and hit more and more shrimp farms around the world. The white spot virus has a devastating impact on shrimp farmers as it can wipe out a grow-out pond within 7 to 10 days. Multiple challenge tests demonstrated significant mortality reduction from white spot by oral delivery of our product prototypes. These successful trials triggered a joint development collaboration with Stretting and Otreco company towards product commercialization. As cost of production is a major part of the challenges facing the RNA-based solutions, we've invested a large proportion of our resources towards the development of scalable production process and have successfully reached a full pilot scale. Our production process is fermentation-based, and our proprietary downstream process has been developed to include the formulation step, which allows cost effectiveness required for the aquaculture market. Our platform has the potential to provide a suite of solutions for the aquaculture industry, starting with double-strand RNA to shrimp for viral disease management. This strategy is based on triggering a cell mechanism that silences viral genes, the RNAi mechanism, but also oral delivery of a variety of RNA and DNA products as treatments and vaccines for the fish industry. These solutions may replace, in some cases, current injection-based vaccines, and may also be used as oral immune boosters for available vaccines to improve their effectiveness through the entire fish life cycle. Our business strategy involves partnerships with key strategic leaders that will take our developed products to the market. Our strategic partner for the first product is Stretting, one of the world's leading aquaculture feed companies. This partnership provides the Aqua worldwide marketing and distribution network via Stretting's global infrastructure. Here you can see our diverse experienced leadership team with complementary skills in molecular biology, marine biology, drug delivery, business and senior corporate management. And here the presentation ends, and thank you very much for your time. And I'll be happy to answer any question during the Q&A session and afterwards, of course.
Thank you, Shai. I would like to request for Shai to stay on screen while we invite Tarja and Dr. Marcella to join us for a short Q&A. Once again, in the interest of time, I'll ask each of you one question. So for Tarja, with the increased use of RAS systems, do you see a change in the disease situation and the need for vaccines? Tarja, you're on mute.
Sorry about that. Okay, thank you. That's an interesting question. For salmon, we see that that fish tend to be kept for a longer time on land before it's transferred to sea, and some of the fish are vaccinated at a bigger size than before. With recirculation systems, you are able to control the environment better compared to, for example, cage farming in lakes or the sea. But the fish will still be exposed to pathogens, and outbreaks can be severe in a closed system. And also, it's not unlikely that the flora of pathogens will be different in a closed system compared to in a lake or in the sea. But there's limited knowledge about this so far, and in Pharmac, we are very interested in collaboration with farmers that use this kind of technology in order to better understand potential new disease challenges when using this type of technology.
Thank you very much, Tarja. My next question is for Dr. Marcella. Is genetic selection the true remedy to control shrimp diseases?
Yes and no, Ethan. I think, as I show you, genomic selection and genetic breeding is a key tool in preventing diseases in animals, but it's not the only tool. And I think that all the farmers must be aware that the best is management practices. We have to keep the animals under the best conditions to try to prevent diseases, and also trying to work with health surveillance and identifying the diseases early. It's not only relying on genetics that will solve the problems. Genetics is a tool that should be used only when management procedures do not work. It's not the only one.
I see. Thank you very much, Dr. Marcella. My last question for Shai. What is Aqua doing differently over its competition that enables it to bring the technology quickly to the market?
Okay, so I think that our main competitor's solution is based on live genetically modified organisms. And our product is a formulated active molecule, which is more based on industrial biotechnology processes. And we think it's making the effect of the product more predictable and stable than the effect of a live organism that produces the active material. This is one point. The other point, I think that related to the environmental issues, when using a live organism in feed, it makes more concern about environmental impact than when using a formulated product like we do. So it also will allow us to reach, I believe, the market faster than the other solutions.
Thank you very much, Shai. So we've come to the end of the webinar. So thank you to all our speakers for your active participation today. We hope that you have benefited from the insightful presentations and hope they can help generate some ideas about aquaculture innovation for you.